Hello dear students, till now we have seen how exactly the formation of vesicle takes place as well as fusion of vesicle is going to happen. So in this particular class of the video we are going to study about this particular vesicle transport, and then we are going to discuss about how exactly the vesicle formation and fusion is going to play a role in the process of movement of molecules or proteins within the cell. So first of all, I wanted to explain and discuss about the study of importance of the Golgi complex in the process of glycosylation or protein modification. So this particular process or importance of vesicular transport, you can see how the mechanism of transport of vesicles also was studied in great detail by using a simple experiment wherein people have taken the Golgi complex. So wherein they have taken a Golgi complex which is having mutation for N-acetyl glucosamine transferase 1, because of which it is unable to transfer the glucosamine during glycosylation. So, and they also have taken a protein means the viral protein which is need to get transferred with this N-acetyl glucosamine in order to be very much functional. So what they have done is they have taken the Golgi complex, they incubated the Golgi complex along with the together they have added little amount of cytoplasm as well as ATP and N-acetyl glucosamine, which is tritiated, radio labeled, and then they incubated with only the Golgi complex which is mutant, and what they found is with that particular mutant Golgi complex there was no uh, protein glycosylation because of that mutation in the enzymes. And then after doing that, so what they've done is, because this particular now these Golgis are having the viral protein molecule, but these viral protein molecules are not completely functional, and these viral protein are found to be the protein present within the lipid layer of the virus. So now what they have done is they have taken an acceptor Golgi complex, or in other words the Golgi complex which is wild and not mutated. Now they have added this few of the Golgi complex, and this particular protein what they have taken, the viral protein molecule, was tagged with green fluorescent component. So it was a fusion of green fluorescent protein. So thereby they could able to see how exactly this movement of protein even it takes place within the Golgi cisternase. So when they added this particular acceptor Golgi complex with the mutated donor Golgi complex along with the cytoplasm, ATP and tritiated N-acetyl glucosamine, the UDP linked one which is required for the transferase activity. So what was identified is there was increased consumption of ATP and cytoplasm was also there. So and there was a increase in the radio labeling was observed. That means the tritiated N-acetyl glucosamine now, so initially the acceptor Golgis were not having any viral protein molecule in them. So that means there was no fluorescence as well as there is no radiation and even no fluorescence because no protein in that. So once this acceptor Golgis added into the mixture, what they found is, so there was a radioactivity was seen in the acceptor Golgis as well as fluorescence was also observed in the acceptor Golgi as time increases. That means by somehow from the donor Golgi the protein might have got mobilized into the acceptor Golgi. So when it was observed, they noticed, you can see in this particular fluorescent diagrams. So initially it was there only in the acceptor Golgi. Now slowly the fluorescence is going to start moving into the donor Golgis, and thereby it shows that through the, what I can say, uh, by somehow there should be transfer of this particular protein from the donor to the acceptor. That is only possible with the formation of vesicle. So that's how people actually come to conclusion. So vesicle formation is going to be plays important role in the transport of protein. And here, wherein all this vesicle formation and vesicle is going to be playing a role in transport of proteins, you can see in the phagocytosis there will be formation of vesicle. Phagosomes are going to get formed, which is going to transport that particular phagocytized bacteria to the lysosome. And even during the process of endocytosis, you can see on the other hand there will be formation of early endosome and late endosome. So that is also again formation of vesicle, and that late endosome fuses with the lysosome, that is fusion of vesicle. On the other hand, during the process of phagosomes, so again in the formation of phagosomes also, the lipid membrane layers are going to come and surround and form a vesicle, and then they are going to engulf the whole organelle, and that vesicularized autophagal organelle, autophagosome, is going to again go and fuse with the lysosome and degradation will take place. So not only this, the vesicles are also going to be required. See, in all these cases there is a movement of vesicle is taking place, and these vesicles are carrying the cargo in order to move the protein or the content into the lysosome. Similarly, they will also going to be involved in the movement of different varieties of proteins into the specific organelle, as well as during the secretion of the proteins to the through the cell membrane also they require this particular vesicles. So I'll quote here an example of protein which is going to be functioning within the lysosome. So the lysosomal protein molecules are going to get transported or mobilized into the lysosome through the vesicles. So this mobilization of proteins from the site of its synthesis to the site of action. So because we all know lysosome is going to be the house for many of hydroxylases and many of the proteins which are many of the enzymes which are going to involve in the cleavage, oxidases, catalases. So many of these particular enzymes are going to be there within the um, uh, what I can say, so the lysosome, especially the hydrolases and hydroxylases, they are going to be mainly housing there, which involves in the hydrolysis process. So these particular proteins, even lipases, so these proteins are going to get mobilized into the lysosome, and many of these particular hydrolases are going to be the membrane protein molecule. So whenever we think about a membrane protein, when we think about a protein synthesis, it happens at the endoplasmic reticular membrane, the rough endoplasmic reticular membrane, through the co-translational insertion process which we discussed earlier, and then followed by that they'll get transported from the endoplasmic reticulum into the Golgi cisternes, wherein cis Golgi, trans Golgi, medial Golgi, trans Golgi, they'll all move through that, undergoes all the kind of protein modifications. Once all the modifications, everything is done, now the protein is ready to be functional. So this particular protein need to be transported into the specific site of action. So if you remember the previous slides wherein I have actually shown you the table where it was having the specific signals which actually says decides the destiny of the protein molecule, or in other words, the site of action of the protein is encrypted in the form of a specific sequence of amino acids, or sometimes through the modified amino acids. So they are going to be encoded into the protein molecule, so which will read by various kind of receptors, by various kind of protein binding other proteins through the protein protein interaction, and then these recognized addresses, or the encrypted sequences are going to be the signal sequences are going to be read, and then based on that these proteins are going to be mobilized into the specific destinations. And here this particular mobilization is going to happen in, I'm going to specifically concentrate on mobilization of the proteins through the vesicles. So here in case of lysosomal protein, this particular mobilization is going to happen by reading a sequence of amino acids, and not specifically, but in turn it is going to be through and modified sugar, that is mannose 6 phosphate. So a protein which is going to have a oligosaccharide unit or a glycosyl moiety having terminal mannose 6 phosphate residue is going to and signal that that particular protein is destined to work in lysosome. So now how this particular addition of mannose 6 phosphate takes place during glycosylation is, because it is a addition of phosphate group should happen at the sixth position of the mannose, but generally you can say it is by through simple kinase reaction by the gamma phosphate of ATP can be transferred onto the mannose to form mannose 6 phosphate. But that's not the case here, as the cell is going to be doing things very regulated in a very regulated fashion, and whatever the substrate used for the process of glycosylation, usually they are going to be nucleotide diphosphates or monophosphate sugars. So here how this mannose 6 phosphate is going to get formed is, the protein is going to have an oligosaccharide unit with the terminal mannose residue. To that mannose residue, an enzyme called as N-acetyl glucosamine phosphotransferase enzyme. So this enzyme is going to transfer the uridine N-acetyl glucosamine, that is UDP N-acetyl glucosamine will be taken as a substrate by this enzyme, and it will transfer N-acetyl glucosamine phosphate. They transfer N-acetyl glucosamine phosphate to the sixth position of the mannose and gives out the nucleotide UMP. Now after adding this N-acetyl glucosamine phosphate to the mannose at sixth position, followed by that this phosphotransferase enzyme, what it will do is, now it is going to show its phosphotransferase activity and removes the N-acetyl glucosamine. You can see here it removes N-acetyl glucosamine. By the removal of N-acetyl glucosamine, it is going to produce what I can say, the mannose 6 phosphate residue at the terminal of the oligosaccharide unit attached with the enzyme. So this particular protein now is going to get recognized uh, by the receptors of mannose 6 phosphate, and they bind with this particular protein. All this whole process will take place in the cisternase of Golgi, and in the trans Golgi network this mannose 6 phosphate residue will get attached with the protein called mannose 6 phosphate receptor. So this formation of mannose 6 phosphate happens specifically on the hydrolases which are going to be destined to function within the lysosome. So you can see here how whole the process is going to happen with the N-acetyl glucosamine phosphotransferase enzyme. The protein with the lysosomal hydrolases which are going to be found within the Golgi cisternae get again transferred with this UDP N-acetyl glucosamine, then UMP comes out, then followed by at last N-acetyl glucosamine is going to get removed, leaving the phosphate at the mannose at its sixth position. So once the mannose 6 phosphate get formed, now this is going to be said this protein is need to be functions within the lysosome. So how this particular protein mobilization happens from the Golgi, that is trans Golgi network, because that's called as sorting station, from where the proteins are going to get sorted. Sorting means it is distributing, or in other words separating the protein molecules based on their signal sequences available on it, or the signals available on the protein, and making sure that this particular protein is mobilized into the specific address, or they are going to be transported or moved into the specific address. So this is going to happen in case of this hydrolases in two different pathways. One, we say it's going to happen through a secretory pathway, another through the endocytic pathway, one through endocytic pathway or secretory pathway, the other is going to be through the simple transportation path. So here the protein is going to get transported. You can see that in the trans-Golgi network, the protein with the phosphate is going to bind with a you can see the orange color receptor, that is a mannose 6 phosphate receptor. So this particular receptor is going to bind with the proteins which are having mannose 6 phosphate at its terminal. So this particular receptor is going to be there on the, means it's going to be present at the membrane of trans-Golgi network, and once the protein binds with it, we know through the clathrin coated cotomers, there will be formation of vesicle. Here it takes place and forms an clathrin coated vesicle, and later this clathrin coated vesicle is going to undergo the uncoating and forms a transport vesicle. So this particular transport vesicle later get transported through the cytoskeletal element, specifically into the lysosome through and formation of late endosome. See, the transport vesicle, the pH is going to be around neutral, but later with the pumping of H plus ions into the endosomes, it becomes acidic pH inside the late endosome. So that forms a late endosome where low pH is going to be there, and because of reduced pH, there is acid hydrolysis will happen, where this means the acidic condition is going to reduces the affinity, or other words it's going to release the interactions between the mannose 6 phosphate having hydrolase and the receptor, and followed by that the phosphate is also going to get acid hydrolyzed and get releases this particular hydrolase free into the medium. So then what happens is that the mannose 6 phosphate receptor which was there on the membrane of late endosome is going to be further again undergo vesicularization through the clathrin, and that receptor, which is without any kind of ligand, is going to get vesicularized and further undergoes uncoating and again become transport vesicle, and this transport vesicle can go and fuse with the trans-Golgi network, or even it can go and fuse with the plasma membrane, because the membranes are also going to found to have these receptors. So now this late endosome which is going to get formed, this late endosome is going to now fuse with the lysosome, and thereby it's going to make sure the protein is going to get mobilized into the lysosome. So this is one particular pathway through which, at the intracellular pathway, wherein the lysosomal protein is going to get transported into the lysosome through the formation of vesicles with the help of mannose 6 phosphate receptor. The other method is simply this particular lysosomal protein can get transported into sorry, here the phosphate is missing, so it's going to be an lysosomal protein hydrolases so which is going to get again transported, means transported as a secretory vesicle, wherein again formation of vesicle takes place at the trans-Golgi network, and it goes and fuses with the plasma membrane and pushes out the protein into the extracellular side. So now at the plasma membrane, as I mentioned earlier, the mannose 6 phosphate also going to be present on the plasma membrane. Now this particular protein which got secreted will get attached to this receptor, which further induces the formation of an receptor mediated endocytosis process, wherein the so there will be receptor mediated endocytosis, wherein the receptors, that is mannose 6 phosphate receptor, binds with the ligand, that is the here the hydrolase protein enzyme, and then which induces the formation of a coated pit, that is the clathrin coated pit, and then further polymerization of clathrin will happen. There will be formation of a coated vesicle which gets uncoated and becomes transport vesicle, and then that's the early endosome, we can say it as, and that early endosome with the change in pH becomes late endosome, wherein again the hydrolase is going to get away from the receptor, and even hydrolysis of phosphate will takes place, and late endosome fuses with the lysosome, wherein the receptor is going to get recycled back into the trans-Golgi network or into the membrane. So this is how the lysosomal protein will get transported into the lysosome through the formation of vesicle and fusion of vesicle, which is going to be mediated by the clathrin protein molecule. So you can see this is electron micrograph, wherein there are many number of proteins that are cargoes are going to get transported. Here it is LDL and ferritin, which is being shown here. So another important process which we come across during this particular method is receptor mediated endocytosis. So receptor mediated endocytosis is nothing but it is a process in which the specific receptors present in the plasma membrane are going to bind with the ligand. So the interaction of ligand with the particular receptor can cause the receptors to undergo what I can say, it's going to cause this particular conformational change within the receptor, specifically at the cytosolic side, which exposes the conserved sequence of amino acids which signalizes for the endocytosis, like uh, YXX5, that is tyrosine XX5, so which is one of the type of sequence which is going to be present on the cytosolic of the receptors. So upon exposure of which is going to be bind with an adapter protein and as well as the ARF protein molecule, which induces the formation of clathrin coated vesicles, and then there will be uncoating and the vesicle formation will take place. So here the vesicle formation in case of receptor mediated endocytosis happens at the plasma membrane. Okay, so thereby the particular ligand is going to get endocytosized and will get transported to the lysosome. There is also one more method of endocytosis which is called just simple endocytosis, and this endocytosis also happens by the formation of coated vesicles, but here the at the plasma membrane it's not that always there will be means the not that always the vesicle formation is going to get induced, and then only the clathrins are going to come and polymerize and forms the vesicle. Sometimes pre-polymerized clathrins will be present at the membrane, wherein they are going to be present as a coated pit. Coated pit means at their cytosolic side the membrane is going to have the coating of already clathrin proteins, and which is going to be ready to interacting with the receptors. So this also can happen that sometimes the proteins are going to bind with the receptors at somewhere in the plasma membrane, and as the plasma membrane is dynamic in nature, it will get flows within the membrane and reaches this particular coated pit, and then the endocytosis can happen from this particular coated pit. So like that also the endocytosis will takes place, and happens that means it will bring about the movement of molecules into the cell. So here I am going to give you a brief insight about the LDL endocytosis, which means undergoes receptor mediated endocytosis. So LDL, which is nothing but low density lipoproteins, which is going to be transported within the blood. So this LDL is going to get formed at the liver and get transported into the adipocytes. And whenever the cell requires more energy and requires this fatty acid to be get catabolized, again from the adipocytes these molecules are going to get formed. The LDLs can get formed and again get transported through the circulatory system blood into the liver, and then can undergo metabolism. So this so here uh, in this case of LDL, once again this LDL is like this. You can see the outer lipid layer having an apolipoprotein followed by, that is, apolipoprotein B, and inside the LDL there will be a polar core where it consisting of all these lipid contents as cholesterol esters in cholesterol, as well it can have triglycerols and all these things. So this LDL receptor is a 839 amino acid residue having glycoprotein present at the plasma membrane, having single transmembrane segment and a cytosolic short chain. So this exoplasmic side, this particular segment is going to be very key here. You can see here this is going to have a beta propeller domain. Beta propeller domain is nothing but it is a cysteine rich 7 cysteine rich imperfect repeats from the ligand binding side, so which is going to act as beta propeller domain. With the beta propeller domain, this is going to interact the LDL and binds with that particular ApoB protein. So you can see here the interaction, how it happens at the cell surface. So this is how this particular protein is going to bind, and there will be NPXY sorting signal at the cytosolic side, and this NPXY sorting signal is going to get recognized by the adapter protein molecule, which results in the formation of uh, coated vesicles. You can see here the LDL carrying the B100 or apolipoprotein interacts with the receptor, which is already present within the coated pit, or it may be present freely and then it can get transported into the coated pit. That is also possible. And once it is going to get transported with means once it is going to bind with the coated pit, further there will be uh, formation of vesicle happens through the clathrin polymerization. Uncoating of the vesicle will first coated vesicle will get internalized, and then uncoating will takes place. That uncoated vesicle undergoes changes wherein pH is going to get reduced, so which releases the LDL from its receptors in the late endosome, where the pH is going to play an important role, and then the receptor portion of the late endosome is going to get vesicularized, means forms a vesicle, and results in the formation of the transport vesicle. So that late endosome portion is the carrying the LDL will get fused with the lysosome. On the other hand, the vesicle carrying the receptors will go and fuse with the plasma membrane, through which it recycles the receptors, and on the other hand the lysosome is going to hydrolyze the LDL into the its components, that is into the amino acids, cholesterol and fatty acids, which further utilized for oxidation purpose. So these receptors again they will be ready for one more round of endocytosis. So that's how the same thing here, that's how the endocytosis of the LDL will takes place. That happens through receptor mediated endocytosis. There are a few more examples for that receptor mediated endocytosis which we come across with here a protein that is transferrin. So this transferrin is a protein which carries the iron. So this transferrin exists in two forms, apotransferrins and ferrotransferrin. Apo, which is not bind with the iron Fe3 plus, so and ferro, which is going to bind with the Fe3 plus. So this ferrous ion binds with the transferrin, because transferrin is the protein which actually involves in the mobilization of iron and facilitates the movement of iron to a larger extent within the body. So this particular iron binding protein binds with the iron, two ions of iron, and then it has a specific receptor called as transferrin receptor. So it interacts with the transferrin receptors and forms a receptor mediated endocytosis, wherein there will be formation of coated pit, followed by that the vesicularization happens through the coating, coated vesicle get formed, then uncoating happens, then get converted into early endosome, late endosome, wherein the transferrins get released, the transferrins are going to unload uh, the ferrous ion, whereas Fe3 plus ion, whereas the transferrin with the receptor is going to get converted into vesicle, and that vesicle is going to get infused with the plasma membrane, and once it fuses with the plasma membrane, so there the release of the apotransferrin will takes place from the receptor, and all this happens with the change in pH. Okay, and that Fe3 plus, which is going to get means which will be left out in the late endosome, will further get utilized wherever Fe3 plus is required for the reactions. So this is how the transferrin is going to be playing a role in movement of iron, uh, and which is facilitated through receptor mediated endocytosis. So this is another example wherein autophagosomes are going to be there. Autophagy also requires it, uses the same process. So in autophagy, you can see the peroxisome is going to get autophagocytosized by the vesicularization which happens through the lipid layers. So some of the lipid components, vesicle components which get produced at the endoplasmic reticulum or at the Golgi complex, they will also act as a phagocytic and autophagocytomes. They go and encircles the organelles and forms autophagosomes, which again get transported and fuses with the lysosome, and lysosome further degrades the particular organelle. So this is how rabies virus, that is viruses, are going to get endocytosized through receptor mediated endocytosis. This rabies virus is carrying a ligand on its surface, so a glycoprotein molecule, this will get recognized by the receptor on the surface of the cells, and then endocytosis will happen, and then that endocytic vesicle, which is uncoated endocytic vesicle, is going to have again a fusion, wherein the viral envelope is going to now fuse with the membrane of an endosome. So earlier this is an enveloped virus, so here this enveloped virus is going to fuse with the membrane of endosome. Okay, it can fuse with the plasma membrane, but it first looks for a favorable entry and protected entry. Once it gets in, fuses the endosomal membrane, it releases the viral protein with the RNA, which further undergoes transcription, gives protein, and these proteins are going to be gets in mRNA, which gets converted to proteins on the endoplasmic reticulum. Okay, and even that RNA undergoes replication and gives number of copies of its own, and even also codes for varieties of protein. So all these proteins, and as well as proteins from the matrix, from the endoplasmic reticulum, so now that can combine together, work and establishes the formation of the Golgi complex with the viral glycoprotein molecule, and then further from the Golgi the assembled viral genetic material as well as the receptor proteins along with the membrane. So the functional viral molecule is going to get exocytosized, or in other words I can say they get secreted. So the secretion here, it's not exactly secreted, it's going to happen through a kind of budding. So where this particular proteins, the proteins of the membrane is going to get studded into the membrane through the vesicles, and then the portion of the membrane where the viral envelope proteins are going to get studded. So at the particular same site, the progeny virion, which is an assembly of the nucleoprotein, assembly of capsid and capsid protein with the genetic material, that is RNA, together it forms nucleoprotein, which again now get transported at the membrane which is studied with the envelope protein, and then the portion of the membrane is going to get extended and gets budded out and give rise to a particular region. So whole the process will take place at a very small stretch of time, and that leads to the transfer of or the uh, entry and exit of this virus from the in the case of cells. So here similarly the HIV, you can make out HIV also going to infect with the similar kind of mechanism, but here it is the fusion of the viral envelope will happen with the plasma membrane, and which is going to allow the entry of the nucleoprotein into the cytoplasmic site. So in rabies virus it is the whole envelope with the protein where the nucleoprotein will get inside, and then the release of the nucleoprotein happens by the fusion of envelope with the endosomal membrane. But in the case of HIV it is direct fusion of the membrane of the target cell with the envelope of the HIV virus. So that directly binds there, and then it infects, where multiplication will happen, and then checks whether everything is fine or not. So then after once everything is there, everything is ready, then again viral proteins are going to get studied in the plasma membrane, again budding, through budding the newly formed viruses or virions are going to get uh, secreted out of the particular cell. So this is all about receptor mediated endocytosis and few examples, and vesicle formation, fusion, all these things. So I'll just explain one small aspects of transport of vesicles in the next class, in the next video, which you have already studied in the first semesters, wherein we have studied through the cytoskeletal element where kinesins and dyneins are going to be involved in the movement of vesicles or cargo in an anterograde manner and retrograde manner on the microtubule, and followed by that the the myosins which are going to take up the cargo and transports at the membrane at the periphery of the cell on the actin filaments. So this combination of actin and myosin and their motor proteins actually facilitates the movement of vesicles at the inside the cell. So here there will be networking of this particular one, and you can see there will be lot of trafficking is going to be happening, means lot of movements of proteins and vesicles will be taking place simultaneously on these cytoskeletal elements, so which is going to create a huge traffic inside the cell. And this huge traffic, you can see that it will happen so efficiently, the traffic is going to be moved, will be moving so that there will be never a traffic jam. So if there is any kind of jam, that is a traffic jam inside the cell, that means that particular cell is going to be undergoing the process of death, because the traffic jam which has happened inside the cell is going to cause an what do you call it as feedback, or in other words I can say there will be plenty of accumulation of other varieties of cargoes at other different destinations, which will make the cell become inefficient in doing its life activity, and so it will be subjected for death. So that's so much important is this particular process of mobilization of proteins, either in the form of vesicle or in the form of the free form, and then that mainly happens through specific sequence of nucleic amino acids which are going to be existing at the protein in the form of signal sequences, or even it can be in the form of modified amino acids or modified sugars. So this is all about this part of the membrane biochemistry. So with this I can say I have concluded all the aspects of membrane biochemistry which was allotted to me. In case if you find any of the aspects which is not touched upon or which is not discussed, about which you need more information or anything, if you are going to post it to me as a message or something, so regarding that I can make a video and send it across, or if you want personally to be explained you can give me a call, I can explain it over the phone. Thank you, all the best. So from next onwards I'll start with the spectroscopy, and hopefully we'll complete it little more early. So thank you, take care, wash your hands.